Hello, everyone, and thank you for joining us today. My name is Tony, and I will be moderating today's session. With that, I'd like to begin today's conference and hand the call over to Andrea Coffin for our opening remarks. Andrea, please go ahead.
Thank you, Tony. Good morning, everyone, and I would like to invite you to this webinar sponsored by the Annex 4 program. Annex 4 is a task under Ocean Energy Systems, a collaboration of nations interested in the development and responsible use of marine renewable energies. We are fortunate today to have two speakers out of Scotland addressing the role of biofouling in marine renewable energy development. Our two speakers, Dr. Rayanne Miller and Dr. Jennifer Loxton, will pick up where I leave off here. I'm going to introduce Rayanne now and then Jen when she takes over. Dr. Rayanne Miller is based at the Scottish Association for Marine Science in Oban, Scotland, which is a partner with the University of Highlands in the Islands. Rayanne's a benthic ecologist with a background in larval ecology and marine population connectivity. She's a knowledge exchange fellow with the UK's Natural Environmental Research Council, and she works specifically on establishing links between marine science research and offshore renewable energy industries. Rayanne's going to highlight areas where knowledge exchange between biologists and industry can help understand how biofouling communities develop and whether their makeup and potential consequences for structural design and survivability are predictable. Specifically, she's going to focus on the environmental factors that influence marine growth communities, the outcomes of a recent industry consultation and workshop on biofouling, and these particular events highlighted key quick wins for both research and development in this area. So I'd like to turn it over to Rayanne.
Thanks very much, Andrea, and thank you to everyone for being online today and coming on for this webinar. As Andrea has said, I'm going to talk a little bit about biofouling, and the first bit of this webinar is up to me to really give a bit of an introduction. And what I'm going to talk a bit about today, first of all, is just a bit of basic information about biofouling. And we've got a broad audience, so just trying to give everyone a similar background. I'll talk a little bit about legislation and guidelines, but then really get into the nub of why it's an issue, as well as the biology and a little bit about some of the key species that we think might be important for the renewable energy industry. I'll talk a little bit then about how it can be prevented, and also just a note that this isn't an issue that only occurs in renewable energy, but that it's also something that's experienced by pretty much all marine industries. So I'll start off talking a bit about biofouling. Now, biofouling is generally considered to be any unwanted growth on submerged structures. So I've got a few different examples here. What you see in the top left is a submerged ship's propeller on a shipwreck, and you can see it covered in lots of nice green growth. In the top middle, we've got a set of wind turbine support structures covered in mussels, and on the top right is a navigation buoy, again covered in different types of kelps. And you see again in the bottom three images other types of marine growth on man-made structures. And I just want to make it clear that today I'm going to talk about macrofouling, so that's larger organisms which are mostly visible to the eye, as opposed to microbial and bacterial films. And microbial and bacterial films are what are initially formed on submerged structures and allows these larger macrofouling to grow on the structures. And just to illustrate that pretty much anything that gets put in the sea for an extended period of time will get biofouling on it. This is an example. This is our mooring, which we deploy here at SAMS in the Tyree Passage off the west coast of Scotland. This mooring has been in the sea for about 18 months to up to 2 years, and what you see is it is absolutely covered in kelp. And again, that's just after 2 years. So pretty much anything, including renewable energy devices, will get covered. Again, these images are courtesy of a wave energy company here in Scotland called Albatern. But what you see is their squig device, both submerged at the top and out of the water at the bottom. And you see it should be yellow, but it's brown. And this was after a short-term test deployment. And this growth is starting to develop, and if left in the sea, it will develop more and more. So just a brief little bit, I'm a biologist, so I had to get some biology in here. But most biofouling creatures are predominantly filter feeders. And so that means that they need a flow of water going past their habitat or the structure that they live on that they filter food out from, and that's what helps them to grow. They also have planktonic life stages. So what that means is they've got a solitary phase, the nice anemones that you see on the left-hand side are the solitary static phase, but they also have larvae which they extrude into the water column and which float about from place to place until they find a new habitat to live, and then they settle down and grow and turn into adults. Now, each of these life history stages are dependent on a number of different abiotic factors, so that means things that have nothing to do with biology, things like water flow, wave exposure, light availability, and biotic factors. So that's things like competition for space, as well as many other environmental variables. So just a bit more of an example here. I'm a barnacle biologist specifically, so I had to include some barnacles somewhere. What you see at the top here on the left-hand side is some barnacles, adults in their habitat, and they release their larvae into the water. That's in the middle, the thing that sort of looks like a spider. And then it carries on in the water column and turns into a cyprid, the oval-shaped thing that you see. And during this time, it's floating about from place to place, influenced by the currents and water flow patterns, as well as environmental characteristics. And eventually, that little barnacle larvae finds another rock. It attaches itself to the rock and then grows up and becomes an adult barnacle. Now, at the bottom of the screen, you see some wind turbines with an arrow between them, and that's just to indicate that it doesn't matter what type of habitat or structure it is. The exact same process happens whether it's a natural or an artificial habitat. It's all the same to a barnacle. And in turn, the environmental conditions that affect where the larvae go from place to place will also affect the types of communities which grow on the different structures and in lots of different locations. So here, this is just an image of a number of different types of biofouling communities. These come from a paper written by my colleague, Adrian McLeod, and he looked at biofouling on offshore navigation buoys. So these are floating structures offshore around the coast of Scotland. And what you see is that these communities are really very different. So for example, in the top left, we've got a community that's got a high density of caprellid amphipods, whereas in the middle, on the top in the middle, you see lots of anemones and mussels, and in the top right, you see that there's lots of mussels, and pretty much only mussels. So again, just to highlight here that the different types of communities really depend on the location of the structure or the location of a renewable energy device and the environmental conditions that surround it. So let's look at what might grow on an actual structure. So what you see here is a wind turbine piling. And just to illustrate that the different types of biofouling can also depend on the depth of a structure in the water column. So if a structure is in the intertidal zone, and it's fixed so the water can go up and down on the structure, you tend to get things like barnacles and algae. Further down, just below the surface of the water, where there's still light penetrating the water column, we get kelps, barnacles, and again, different types of algae. And again, going further down, we've got the main column and all sorts of different creatures living there, a bit more diversity with things like anemones, soft corals, tube worms, and again, this really will depend on the depth of the water column. And finally, down at the base, we've got environmental effects, things like scour. Piling may have scour protection around it, and the seabed may have differing degrees of mobility. So again, all of these different things will affect the type of biofouling that we see on different structures. And of course, that's going to be very different between an offshore floating wind turbine, a tidal turbine, a nearshore wave energy device. These will all experience different types of biofouling. So I just wanted to highlight, before getting into the really nitty-gritty, some of the legislation and regulations around biofouling. So a number of different industry standards are in place to regulate how marine growth is accounted for, both in terms of engineering design as well as in structural maintenance, and that's really aimed at the engineering side. And some of the examples include things like the DNV, the Det Norsk Veritas standards for the design of offshore wind and their new tidal turbine guidance. But you can also look at guidance from a number of other industries as well, so things like BMT Corda's guidelines for oil and gas, as well as the International Organization for Standardization. So there's a lot out there already. And just to highlight really that biofouling is not a new issue. So biofouling communities are also interesting in terms of things like invasive and non-native species, particularly in the UK and the EU. Maybe I need to split those now, I don't know. But legislation around that topic's really emerging. And just to highlight that the couple of things that I'm going to talk about are UK or EU examples. But again, this is an international issue. Things like invasive species get all over the world. And the engineering design, engineering requirements, while the biofouling species may be different, it's something that all maritime engineers have to deal with. So a little bit of a closer look at some of the, it says legislation at the top, but these are actually guidelines for biofouling in terms of engineering. And this comes from the DNV guidance for tidal turbines, and it's really just an example to illustrate what kind of guidance is out there. So for example, what you see here is just looking at advisory thicknesses for marine growth or biofouling. And you'll note that these are really based in the North Sea and the Norwegian Sea, which aren't necessarily areas that are relevant to potentially offshore wave energy devices and some of the other marine renewable devices that we see. So this is really an area that we're quite interested in working on a bit, is how are these standards different, or the guidelines different for locations which aren't the central and northern North Sea, or which aren't the Norwegian Sea. And again, just a couple of examples of legislation in the UK and the EU around invasive species. Really because that for devices which need towing from place to place, ballasting, deballasting particularly, invasive species could be quite an important concern. So for example, in Scotland, it's now illegal to accidentally transfer or spread a non-native species as a result of inadequate biosecurity. So just to take that a step further, companies may now be liable for the costs associated with the eradication or control of that species, as well as for restoration of the environment. So that could potentially be a substantial risk, but again, it's something that we can take measures to understand and to mitigate or prevent. So also, just to highlight that at the EU level, invasive species is managed under EU regulation, and that covers both terrestrial and marine environments. But it's also a high-level descriptor of good environmental status in terms of the EU's Marine Strategy Framework Directive. So that's non-indigenous species introduced by human activities are at levels that do not adversely affect the ecosystem. And just to highlight as well that there's a number of other guidelines out there, and these are all things that are pretty much readily found on the internet. And that's things like the IMO guidelines on control and management of ballast water, on management of ships biofouling. And again, there's some advice that can be gleaned there and can be particularly pertinent for the renewables industry. So that sort of leads to the question of, was biofouling actually a consenting issue? And I'll be the first to put my hand up and say, you know, in comparison with some of the issues around macrofauna, megafauna, things like collision with tidal turbines, and some of the other issues that we hear a lot more about, it isn't really a consenting issue, particularly around invasive species. But again, with changing legislation, there's a real need to demonstrate that reasonable steps have been taken to prevent non-native species spread. And that might take the form of something like a biosecurity plan, which includes information about how likely a species is to spread or invade, as well as some of the socioeconomic and environmental risks of spread associated with those key species. So again, not a key consenting issue, but certainly something that should be on people's radar. So now that I've said that, well, why are we here in essence? So why are we interested in studying biofouling? And these are just a couple of examples. Things like biofouling can accelerate corrosion on structures, can cause pitting, as well as changing the loading and hydrodynamic properties of structures. And often that comes along with operational or financial implications as well. So just a few more examples, and these are just things to kind of keep in mind as we move forward across the rest of this webinar. Biofouling can alter the weight of structures considerably. Of course, that will depend on the type of organisms present, what their density is. The thickness of biofouling can affect the diameter of components, and again, that's got knock-on effects on the structural drag and the added mass coefficients. So these all really have to do with engineering design. Furthermore, it can increase the surface roughness, so where hydrodynamics are really important. And again, there's implications for efficiency and drag. And the final one there, biofouling of things like heat exchangers can change the heat transfer efficiency and cooling of a device, so things like gearboxes, etc. So what I thought I'd do in the first part here is just talk a little bit about some of the species of concern. So we've identified these as the different types of organisms which have the greatest effects on biofouling thickness, roughness, and mass and drag. So what you see here in the three pictures is on the left, kelps. Again, that's our mooring completely covered in kelp. Mussels in the middle, and on the right, barnacles. And on the right is a really massive species of barnacles that we find a lot in the Pentland Firth called Chirona hameri. And these aren't just in the UK. These taxa, so these general types of organisms, have been recognized as some of the most common fouling organisms worldwide. So there's a lot to be learned, both in Europe and beyond. So I'll just start with kelp. I thought just to describe for the non-biologists out there just a little bit about each of these groups, as well as the types of environmental conditions where we tend to find them most. So this is now beginning to give us a bit of information that we can use to help predict what type of fouling we might find on different structures in different locations. So we've got a couple of the common kelps here in the UK. Laminaria digitata is the one that we've got here. That is most likely to grow around renewable devices. It's the one that's most resistant to extreme hydrodynamic conditions. So it likes waves and it likes higher tidal flows. But of course, we only find it where there's light. So any structures that are deeper down than light can get, we won't find things like kelp. And of course, they can be really quite big and have some substantial effects on the hydrodynamic properties of structure. And just to mention, I've also heard of a device developer telling me that kelp growing on their device abraded some of their anti-fouling coatings and was having effects that way. And the one thing to remember, though, is that kelp tends to be neutrally buoyant, so that means it doesn't actually exert much mass on a structure. So perhaps not as significant as barnacles or mussels in that regard. Moving on to mussels. So these are found pretty much dominantly in a lot of areas in the upper portion of the water column. So they really like high water flows. They're filter feeders, and good water flows bring them lots of food, so they tend to grow really big. But the thing is with mussels is they've got calcareous shells, which means they're heavy, and they are quite good at increasing the weight of the structure they grow on, as well as the diameter, so the effective size of it. So they've got pretty good effects on thickness and roughness of structures. Now, just an example, in the North Sea, mussels can occupy 80 to 100% of space on turbine foundations and monopoles. By turbine, I mean wind turbines, from the sea surface all the way down to tens of meters in the water column. And that's so much so that there's now actually talk of the mitalization of the North Sea because of their dominance, and how researchers are interested in how that's changing potentially changing the ecosystem in the North Sea. So potentially some engineering issues there, but also some ecosystem things to think about. Finally, it brings me to barnacles, my pet favorite. So barnacles tolerate much higher current speeds and wave exposure than kelp and mussels. So when conditions get really, really, really extreme, that's where we find the barnacles. And that's why we find a lot of them up in the Pentland Firth, where we've got incredibly strong tides. So particularly for wave and tidal devices, these might tend to be a bit more dominant. Now, large species tend to dominate in deeper waters or higher current flows. And a good example is the REDAPT project, which finished up a couple of years ago here in the UK, found the large barnacle Chirona hameri to be dominant on coating test panels in the Falls of Warness. And now these are panels that they were actually testing anti-fouling paints on. And what's interesting, I think, is that this species actually grows faster and larger in areas where they've got the highest flows. So, you know, think of your average barnacles that you find on the seashore, and then think of them being up to 5 centimeters in diameter and 7.5 centimeters in height. And I've heard anecdotal reports that they get even bigger than that. So these can be really big. And again, as a type of hard fouling with calcareous scales, they've got similar effects to mussels in terms of increasing surface roughness and the weight of a structure. So just a quick run over, I guess. Many of you may know quite a lot more than me about this, but there's a number of different methods that are used to prevent or mitigate the effects of biofouling. So biofouling is inevitable, and fouling organisms are pretty persistent, even when things like anti-fouling coatings are applied. And approaches to dealing with this varies quite a lot across the industry and across the wider marine engineering and operations fields. But protective and anti-fouling coatings are used almost universally to reduce marine growth, and the efficiency of these coatings often depends on the environment they're deployed in, as well as on the different types of organisms that are present in that environment. So really what I want to get across here is that not all coatings are created equal. And a number of different projects have tested coating effectiveness. So actually, the photograph on the left-hand side of your screen is from the REDAPT project, which I mentioned previously. And what you're actually seeing is a test rig with a number of different coating panels on it. So they're actually trying to look at the efficiency of different types of anti-fouling coatings. Operation and maintenance plans also tend to incorporate activities to scrape or clean or remove biofouling from specific elements of a structure, and that's really across the entire life of the structure. So not renewable energy, but on the right-hand side is an image of an oil and gas platform riser, which has been taken up to the surface for cleaning. So you can see these guys trying to scrape all of the anemones that have grown on it off. Finally, cathodic protection is another method that people use to control corrosion of structural components, but again, it can be species-specific under certain conditions and can even enhance the growth of some species. So with that in mind, a lot of these techniques are species-dependent or dependent on the type of biofouling communities that might grow. So what really interests us quite a lot is, well, can we predict these communities? How can we tailor some of these prevention and mitigation techniques to a specific community? And again, all of these methods of prevention could really be enhanced with a better understanding of what communities are likely to grow on a specific device in a specific location, as well as with a bit more understanding about which components might be most vulnerable. And this information, again, could help to select anti-fouling coatings and really to develop cost-effective maintenance plans. So I won't go into any of these individual environmental influences in massively great detail. There's a lot of information out there in the literature, particularly for natural communities. Environmental scientists have a really good amount of knowledge about things like currents, waves, salinity, temperature, and how they affect different individual types of organisms. So we understand where kelp really likes to live, we understand which species of barnacles prefer which types of salinity, and so on and so forth. So And more, biologists are using this kind of information to establish which marine communities, so which types of organisms, not just biofouling, but in natural environments as well. So which types of creatures will grow where. So there is precedent for this type of work, and there's been a number of different pieces looking at this in the UK, for example, on subtidal hard communities. But what I want to stress is that it is a somewhat more complex situation than that, particularly for man-made and artificial structures. So these are completely novel habitats in the marine environment. So they're not something that has evolved over evolutionary time. And given that these are novel structures, they offer all sorts of different surface types and orientations for communities to establish on. So you see a number of different types of hypothetical renewable energy devices here. But you see that there's all kinds of surfaces. Some of them are floating, some of them are fixed to the seabed. We've got vertical surfaces, horizontal, ones that will be exposed to the light, ones that won't. And each of these really represents its own microhabitat. And we may even get different communities biofouling on different parts of these structures. So other things to think about might be, is a structure free moving or is it static? Is it floating? Is it fixed? Does it have a splash zone or does it have an intertidal zone? But luckily, these aren't the only structures in the sea, so we're not just looking at renewable energy devices. And we've got a great maritime history, and there's lots of other types of structures that we can make comparisons with. So again, I've said it a few times, but I just really want to highlight that biofouling happens in all industries. Oil and gas, shipping, aquaculture, it really isn't a new issue, and there's quite a lot to learn from these industries. But again, there's a lot of differences as well between the environments that renewable energy devices operate in, the way that maintenance is carried out. For example, getting things out of the water into dry dock may just not be feasible. And again, biofouling really could be particularly relevant for renewables, as devices can be really highly tuned, and any changes in the size of the structure, the weight of the structure, could affect performance. So I'm going to finish there for now, and what I'm going to do is just pass over to Jen, and she's going to tell us a little bit about some more practical information and some of her work on biofouling. So that should be you, Jen.
This is Andrea coughing again. I just wanted to introduce Dr. Jennifer Loxton before she gets started. Dr. Loxton is a research associate at the Environmental Research Institute at the University of Highlands and Islands. And what she will discuss is the implications of specific biofouling communities that are found on marine renewable energy devices in the UK and Portugal and the Azores. The Environmental Research Institute and also the WAVEC Center out of Portugal have surveyed numerous devices, including tethered intertidal and bottom-mounted wave energy converters. They've looked at a tethered tidal device and floating wind turbines. Jen's going to discuss these survey results in relation to potential positive environmental effects and ecological effects, operational and maintenance considerations, and the risk of non-native species harboring and spreading. So over to you, Jen.
Thanks very much, Andrea, and thanks, Rhiannon, for just giving that fantastic background to biofouling. So as Rhiannon said, biofouling is not a new problem. We've got this in all marine industries, but it is something which we're encountering for the first time in the marine renewable energy industry. It's been a fairly long process getting devices to past the design stage and into the test phase. But in the last few years, we've really been reaching that point in Scotland and also in wider places such as in Portugal and further beyond. And we're now at the point where we're having devices being tested for long periods of time, and been fortunate, along with colleagues from WAVEC and from my institute, particularly Chris Null and Ines Mercado, to be able to go out and analyze what kind of biofouling we're really encountering in the real world on these renewable energy devices, And this work's been undertaken under the Marika EU project, which is about 2.5 years in at the moment, and we've got about another 6 months or so to go. So we've still got some more data to come out, hopefully. So I just want to run you through that a little bit. So I'm going to talk to you, first of all, about what are the practical methods out there for analysing biofouling in the real world. And then I'm going to talk to you about the work that we've been doing to establish baselines of biofouling species on marine renewable energy devices. And talk to you a bit about our comparisons which we've been doing between marine renewable energy devices and other man-made structures, seeing how the biofouling varies or is similar. Then I want to talk to you a little bit about settlement panels, which are experiments which we've still got ongoing out in the sea at the moment, so unfortunately I won't be giving you any results from this, but I want to tell you a little bit about the kind of things that you can achieve through these kind of experiments. So to start off with, there's quite a few ways that people study biofouling, from your kind of quite basic through to your more advanced. The most simple method is using quadrats. So this is set size square, often 20 centimetres by 20 centimetres, which will be held on the surface that has been biofouled, and all of the contents will be scraped clean. This can either be done in the water using divers, or it can be done on the surface after devices have been lifted. And at its most basic, this can give you really useful information on biomass. That could be dry, wet biomass, or in water weight. The thickness of the biofouling, and other factors such as rugosity and roughness. As Rhiannon said, these are really useful factors for engineers to understand so that they can actually find out what their devices are encountering in the real world and whether any adjustments need to be made for that. The next type of method is using remote operated vehicles or sometimes divers and taking video footage. So the second picture, which you can see at the bottom of that slide, is some ROV footage. And as you can see quite a lot of detail. This is usually calibrated using something like lasers, which are in the frame, so that you know the actual size that you're looking at, so you can have a look at density and coverage of different species. It does have a few advantages in that it doesn't always involve anyone getting in the water. In fact, usually this is done with ROVs, so it can be done relatively cheaply. You don't need to lift the devices out of the water. And it's been being used for a very long period of time in oil, gas, and wind farm pilings. This is a common way for surveys to be done. It does have a few downsides, however. As you can see, it's quite difficult to see the smaller animals, and it's especially difficult to reliably ID them to species level. And because of this, this makes it a very difficult technique to use for finding whether you've got any invasive species. You don't want to accidentally be identifying something as invasive when it's not really, and it's quite difficult to get conclusive species ID from these kind of images. It's also, if you have very three-dimensional or dense coverage, it can be really hard to really quantify from this kind of footage because you can't see underneath, for example, kelp. You don't know whether or not you've got thick layers of barnacles or mussels underneath that kelp. So it's got its advantages, but it's also got its downsides. Now, the next method is taking scrapes, and this is usually done using quadrats again, so that you're scraping off all of the animals and plants in a fixed area, and you take that back to the laboratory and you ID every species. You can also take the physical measurements, which I spoke about in the first step, at the same time, so you're getting those engineeringly useful statistics. Now, it can involve divers if you're doing this in situ underwater, which is rather more costly, but you can also do this when you're lifting the structure and also from boats. You can usually take scrapes about 1 to 2 meters down using special equipment. However, it does require a bit more skill because you do need to have a laboratory and you do need to have people with ID skills to be able to figure out what all these species are. However, what it gives you is you get a complete species list. and that includes knowing whether or not you've got the presence or absence of invasive species on your device. And when you've got a accurate species list, you can also start to bring in some of the ecology data, which Rhiannon was talking about, and finding out a bit more of this, about the seasonality of settlement for those species and how big you can expect them to grow. So you can start to learn a little bit more, which might be useful for you in planning your device and the anti-fouling regime and considerations for engineering. The last method is using settlement panels. I'm not going to talk to you much about this here because I'm going to come back to that later on and tell you a bit more about settlement panels. But this is, I guess, the kind of deluxe version where you can find out a lot more details, and it's the most thorough method. But I will talk to you a bit more about that later. So to talk to you about the recent surveys which we have done, we visited various devices between 2014 and 2015, and sorry, this map is rather strange on here, but we'll just go with it. So we went to 5 different types of devices. They're really not very clear on this map at all, but we went to 2 devices in Orkney, which were deployed at EMEX test sites, a floating tidal device and a floating wave device. We went to a west coast of Scotland site where we visited an array of floating wave devices and sampled from those. In northern Portugal, we have sampled from an offshore floating wind device, and in the Azores, we visited a coastal oscillating wave column, which is quite different to the other devices, but had been deployed for quite a long period of time, so it was very interesting to find out what was on there also. So the methods that we used for taking the samples, we were using scrape samples. For the majority of these surveys, we were using quadrat and fixed area scraping. There is one exception which I would highlight to you, which is for the floating wave array on the west coast of Scotland. Unfortunately, that had been lifted from the water a few days prior to our sampling, so the scrapes had to be taken in a more opportunistic manner. Because of that, I would highlight that the species list for this device is likely to be underrepresenting mobile species and those that are affected by drying out or desiccation. So just keep that in mind when you have a look at the other results. With the other devices, we used divers, snorkelers. We took some samples from the boat, and some we were able to take freshly after the devices had been lifted. So very, very high-level results. Overall, across all of these devices, we found 217 species of algae and animal. On the floating tidal device in Scotland, we found 44 species. Floating wave device in Scotland, also in Orkney, we found 89 species. The floating wave array on the west coast of Scotland had the fewest species at 27, but this is the one which I would remind you we believe is underrepresenting mobile and soft species because it had been listed out previously. The floating wind site in northern Portugal, we got 44 species, and the oscillating wave column in the Azores also had 25 species, which is a fairly low number. You might notice that the division of species among phyla is quite different also for the Azorean site, which is the far right column there. We have far more algae than any other species. So that was quite an interesting thing to have a look at. So we thought that we would have a look at this data and compare it to other man-made structures in the sea. So we went to the scientific literature to find out what we could from the literature. There were a number of papers out there which had species split amongst phyla. Many of these were in Scotland, but we also managed to find them in other parts of the North Sea. We were fairly light on literature from Portugal and the Azores. There's just not much that has been published on biofouling, but we also tried to find some comparable structures which we could compare to the devices we had down there. So on a map, you can kind of see the distribution a little bit better of where we pulled this data out from. We did find down here in the bottom left, we found another comparable site to the coastal OWC, which we surveyed in the Azores. So we actually compared this to a natural shoreline, because this is a shoreline device, which is quite different to the other ones. In Portugal, we found another man-made structure fairly close to the floating wind device, which we surveyed. And then in Scotland, we were much more fortunate in that we had a lot more data there to work with. So we found oil platforms and also fixed wind farms in the southern part of the North Sea. So with this comparison to other man-made structures, we could ask the question, does location matter? Well, the answer is to an extent, yes. Cluster analysis showed that 3 of the sites in particular were significantly different to the other sites, and we excluded these from further analysis. And this isn't really probably very surprising to most of you, I guess. So the 3 sites that were significantly different were all in, well, 1 was in Portugal and 2 were in the Azores, which is, of course, also part of Portugal. And these were all fixed shoreline sites and devices. So this kind of really explains why they are different. And we also have the difference there of the Portugal and Azorean flora and fauna being rather different to what we find in Scotland and the North Sea. So we excluded those ones from further analysis and then moved on to have a look at what we had from the remaining sites. And we did some principal component analysis so that we could have a look at what kind of groupings we had in amongst this data. Now this is working with the division of species among phyla and seeing which devices or man-made structures are most similar with those divisions amongst phyla to other ones. And we were testing for location, and we were also testing for type of structure. And we divided the type of structure between floating nearshore devices and other. Now that might sound a little bit strange, but one of the reasons that we did it this way is because one of the devices which was behaving rather differently to the other floating nearshore devices was the floating wind farm, which has a much deeper floating structure and is also much further offshore than the other floating devices that we analysed. And lo and behold, when we did this post-fall component analysis, you can see that it's this little light blue square over here, and it is grouped in as similar to the other, these blue upside-down triangles are all wind farms, fixed wind farms. So the type of flora and fauna, the divisions of species amongst the types of flora and fauna, was found to be the same for the floating wind farm and the fixed wind farms. However, one thing that I'd like to highlight to you before we kind of read too much into this principal component analysis is that we have a confounding factor here, which you can probably spot, and that is location. So the main problem that we have here is that all of our floating nearshore devices happen to be in Scotland, and all of our fixed offshore wind farms especially, well, wind farms were in the South North Sea or in Portugal. So really we found that location was confounding this, and we couldn't see whether there was really much of a difference caused by device type. So to try and remove this potential confounding location data, we went on to just have a look at comparing devices within Scotland. So here you can see I've actually put the labels against each of the dots here, which is maybe a little bit more helpful, and we can see the similarities in the communities, biofouling communities within Scotland. And you can see that there's two main groups that have come out here. The one on the right-hand side, you have the Montrose Alpha oil platform, the Beatrice oil platform, and the Heather A oil platform. And you have an extra thing which has fallen into that group as well, which I'll come back to in a minute, which is a floating wave device, the underneath side. So I'll talk to you more about that in a minute. In the left-hand group, we have got navigational buoys, which the information was pulled in from Adrian McLeod's PhD, which Rhiannon was talking to you earlier, and they're found to be in the same group with floating tidal devices, floating wave array, and the near-surface sampling from the floating wave device as well. So we really do seem to have a split here caused by the kind of device. The exception here is these two red boxes, which are the floating wave near surface and the floating wave underneath. So we split this one out because we had the extra data for this particular device. This device is one which many of you may be familiar with. It's the Pelamis device. Pelamis is a floating wave device, which is, you can kind of think of it like a long articulated snake. And it's quite a long device, and it's got quite large floating areas. And Chris Knoll was very lucky to be able to sample extensively on this device, both from near the surface and right the way underneath the device as well. They had, unfortunately, I think most of you will know that Pelamis has unfortunately gone into liquidation. So they are no longer in their existing form. But while they were still testing, they had a rather interesting way that you could get into the connections between these different floating segments, where they could use a habitat where they would pump all the water out, and you would actually be able to access all the way underneath the device and all the way up the sides. So because of this, Chris was able to thoroughly sample, without getting his feet wet, the underneath of the device as well as the sides. And we thought this would be a really interesting thing to look at. And we did find that the near-surface biofouling is statistically different to the underneath biofouling. And while the near-surface biofouling is incredibly similar to all of the other floating devices that we sampled and the other examples from the literature as well, the underneath biofouling is much more akin to what you find on fixed structures such as oil platform pilings. And this was quite interesting and raised the real question as to kind of why. And this seems to be a combined effect of depth and also of light. The underneath of these floats really are kind of out of the photic zone, so they are having rather different structures growing on them. So to talk a little bit about what are the defining phyla. So what are the kinds of groups of phyla that really split these two groups apart in Scotland? So we had a look at the vector overlay analysis from this, and we were able to see that the defining phyla for the oil platforms and also for the Pelamis underside, that exception which strangely fitted in with the other oil platforms, were bryozoans and cnidarians, specifically lots of soft corals and anemones, and echinoderms, so things like starfish and urchins. And we're finding far more of these at these deeper depths and further offshore. In contrast, the other groups that we had, which included the Orkney Sky navigation buoys, the floating tidal device in Orkney, the floating wave array in the west of Scotland, and the Pelamis near-surface sampling, were far more arthropods, including barnacles, far more chordata, predominantly sea squirts. Any of you who have sampled in the UK will be fairly familiar with these. I'm sure lots of other parts of the world get plagued with these lovely squishy things as well. And peripera or sponges. And just below you've got kind of examples of what those look like. That's not to say, of course, that these different communities that we've identified didn't also include algae and mussels. All of these had algae and mussels to some extent, but it's it wasn't a defining phyla because all of these structures had both mussels and algae. So it's really the defining phyla which we're talking here about are the ones which differentiate between these two groups or types of structures. So I also just wanted to touch on invasive species. So with the exception of the pico plant, which I've already spoken to you about, is the coastal oscillating wave column in the Azores. And I said that is a bit of a different beast, so I'm not really going to go into that here. But with the exception of that one, all of the other devices were colonized by non-native invasive species. And the 5 that we found were Caprella mutica, or the Japanese skeleton shrimp, Schizoprella japonica and Schizoprella aurata, which are both bryozoans, which are relatively common. Japonica is in the north at least, and aurata is more widespread across the rest of Europe. Corella eumyota, which is an orange-tipped sea squirt, and Dasyphyphonia japonica, which is an algae. So Rhiannon spoke earlier about the legislation implications of invasive species, but I would also like to highlight that all of these species were also documented in nearby marinas and harbours, and it's incredibly likely that the devices have not introduced these as new species. They've just provided a new habitat in an area where these invasive species already inhabit. So this is really a word of advice and pragmatism when considering invasive species. There is a big difference between providing new habitat in an area which is already kind of colonized by different invasive species and introducing new invasive species. That is not to say, of course, that renewable energy devices don't have any risk at all of introducing new species, but we should look at this pragmatically. One area where renewable energy devices are at risk of introducing new species is the practice of long-distance wet towing, which is fairly common in renewable energy devices. They are often built in other countries and then wet towed to their new sites. Similarly, they are also often ballasted in those new places, and that ballast water can also potentially be a risk. So whenever those long-distance wet towing practices are taking place, it is still important for developers to conduct biosecurity analysis and take any necessary preventative measures to prevent invasive species introduction. So just to kind of summarize this baseline that we've set and what we've found from it so far, broadly speaking, location does matter. We found different kind of biofouling communities with Scotland versus Portugal, for example. However, we didn't find any statistical difference between the different communities around Scotland or in the vicinity of Portugal. The interesting thing which we found is that the biofouling in the top approximately 3 meters of floating structures is quite different to the biofouling on fixed structures or on floating structures but at greater depths below 3 meters. So this is particularly pertinent for devices which have much deeper floating components, where we are likely to see that below those 3 meters, the biofouling that we see is likely to be quite similar to what we've seen before on oil and gas platforms and fixed wind turbines. It's really the top 3 meters of floating structures where we might be seeing something that's a little bit different. And the last point is that invasive species do occur on renewable energy devices, but it's not necessarily a big problem to worry about. They have not necessarily been introduced on the device, and they may already be widespread in the area. So it's really that just that note of pragmatism there to consider what is already in the surrounding area. And really, we would advise before new deployments, it would be great practice for people to set a baseline of what invasive species are in that area before they put their device in the water, so they know what they're likely to encounter, but also so that they can prove it wasn't them. So I'd just like to move on to tell you a little bit more about some experiments which I currently have ongoing and which will continue over the next few years. And this is the rather more detailed study of biofouling which we're doing through settlement panels. So settlement panels are usually about 15 centimetre by 15 centimetre squares, flat squares, often made of hard plastic or some other kind of stable material, but the material can vary. And what they offer above and beyond simple scrapes is you get a really detailed species list, which can be device and or site specific. And this can be more detailed than scrapes because you can look at the tiny larvae which have settled onto the devices, which haven't yet grown to adult size, things which are often destroyed in the process of scraping these off a surface. So you can get a much more complete species list even when you've got early settlements. It also allows really quantitative measurement of biomass, thickness, and rugosity of biofouling. It's quite handy that you can lift these settlement panels whole off the device and take them with you and do all of these measurements, which is much easier to work with than when you're having to do these measurements in situ. A real advantage which they offer is that you can learn a lot more about the seasonality of settlement. You can change these settlement panels regularly. I'm changing our ones every season, approximately every 3 to 4 months. And you can see how the biofouling changes during that time. Not all species settle the same all year round. There are peak settlement periods for almost every species. And it's really useful to understand these and the growth rates of those species as well, so that you can see what seasonal changes you have. This is something which people often don't consider when they're doing things like engineering planning, for example, but especially if your dominant biofouling species is something like Kelps, then that can be incredibly variable in the amount of mass and thickness through the year as it grows larger in the summer and then it dies back. And we get the same with other seasonal species as well. So it can be quite useful to understand a lot more about the seasonality. It can help to inform for a much more intelligently structured anti-fouling strategy. One other thing which you can do with settlement panels, which Rianne kind of showed you some pictures of earlier, is that you can test different coatings. And this is quite useful for developers to actually be able to test the different materials they find on their device, the different coatings which they're considering using, and also different locations. We have found that even at the same site, different locations on the device can biofoul at different rates and with different types of species, depending on whether they're facing into the flow, whether they're on the side or the underside, for example. And this gives you the opportunity to be able to, during the test phase, have a look at how the biofouling differs on these different areas and different material components and coatings. And as I mentioned around seasonality, you can also understand the growth rates, different growth rates. So, so far, we've got settlement panels installed in a few different sites. We have a wave site in Portugal, the Pico plant coastal oscillating wind column device in the Azores. We have a wave test site in Ireland where we have devices, where we have settlement panels out, and a floating wave array in the rest of Scotland. We've also got agreements from developers here in the UK to also put settlement panels in on a new floating tidal device which is going in Orkney, and a floating wind device which will be shortly being put in east coast of Scotland. So when we install these settlement panels, we're usually putting in a minimum of 3 replica plates, and often more, at each site, so that you're really getting some good information there. And they're either being installed on the device or at the energy test site, depending. One thing which is quite useful with these settlement panels is that you can put them in the water in advance of the devices actually arriving at the site. So you can find out about the biofouling and what kind of preventative actions should be taken before anything even goes in the water. So that's something which we've been talking to a number of developers with about doing. As I mentioned, they're changed seasonally, at least 3 times a year. And usually we are installing them in the top metre of water for floating devices, and then installed at varying depths using scuba for either bottom-mounted devices or the deeper floating devices as well. As we found out from the data which we've been getting already, biofouling can vary as you get to different depths, so it's quite useful to be able to get those measures. However, using settlement panels isn't easy. There are a few challenges. The installation method in particular often needs to be different between sites and devices. All of these devices are different, and so what might be practical for one is not always practical for another. And it's also incredibly dependent on the energy of the site, with far more robust fixings needed at more tidal sites, for example, or heavy wave sites. Extremely high energy sites can make installation difficult, and we have much higher risk of experiment loss. This is something we found out to our own detriment, having lost, I think, 3 or 4 sets of settlement panels in the first year that we started to use these due to the high energy sites. So there's a lot of work that needs to be done before installation to make sure that you've got something which is robust enough to deal with the energy of these sites. But I'd like to just finish off here, but I'd like to finish off with a bit of a plea to the people on the line here to say that we're really looking for more sites to install these settlement panels in as well. The more that we can find out about biofouling across these renewable energy devices and test sites, the more useful it will be for other developers and engineers. It's got some useful information, hopefully, for everybody. So if you'd be interested in being involved in settlement panels, please drop me a line. My email address is there. It doesn't look very clear on my screen, however, so it's jennifer.loxton@uhi.ac.uk. Thank you very much, and I will just like to thank my other co-authors, so Chris Knoll, who I mentioned already, Inish Mercado, and also over in WAVEC, Teresa Simas and Erica Cruz, who are incredibly helpful with analysing and collecting all this data. And also a real big thank you to all developers and test site owners who helped us to sample their sites and who are currently hosting settlement panels. We couldn't do this without them. And thank you very much for listening.
Thank you very much, Jen. That was wonderful. And Rayanne, great set of presentations. I have had a couple of notes that people are having trouble with the audio. I don't know, Tony, if there's anything you can check on there. So I hope people are still able to hear. I'd like to get started. We've got a couple of questions submitted here for the speakers, and I'd like to remind people you can type questions in to be answered here today. There's a question for Rayanne that wonders if the reason we see this variety in the different species and biofouling communities can really be parsed out as it would help the wave and tidal sector. In other words, are there timing issues for putting devices in the water, duration that devices are in the water, location, near the surface or mid-water column, geographic location, etc., with the idea that splitting out and sort of parsing these different locations, timing, etc., could help the sector identify what kind of biofouling communities they might have and how to prevent them. Any thoughts, Rayanne?
Yeah, thanks, Andrea. I saw that question pop up. Thank you for asking it. It brought a number of different things to mind. I agree certainly that there's a lot of different things that can influence these communities. For one, I'll just give you an example. People who own a boat tend to take their boat out of the water over the winter. The reason for that is, well, a number of things, the weather in the winter, but also often people notice that if they put their boat in the water earlier in the year, it tends to get covered in things like barnacles almost immediately. Whereas if they put their boat in the water a couple of months later, it doesn't. So, and Jen touched on this a little bit, but many of these creatures have seasonal breeding cycles. So, you know, intuitively to me, the spring and early summer is when we get a lot of larvae in the water column, which means there's a lot of things going in to settle on surfaces. So initially, at least, installing things later in the summer, depending on your location again, but kind of intuitively, you may see less organisms living there. In terms of natural systems, there's also a lot of information out there suggesting that the timing in which a surface is cleared, or even for artificial structures, when an artificial structure goes into the sea, the timing you put it in can actually affect the long-term community makeup. I can't quite remember the outcomes of that off the top of my head, but I'm happy to have a look and do a bit of research and provide further information. Two other things that kind of came to mind immediately as well around that topic. First is there's a paper that I've cited in that presentation by my colleague Adrian McLeod, and he found that actually geography is the most important thing in terms of determining biofouling communities. And Jen mentioned that in her work as well, as opposed to kind of any other physical factors. So it may be that we just haven't sampled in enough places to pick anything out yet, but it may just be that location and the types of larvae that are in the water column are the biggest influence. The last thing that came to mind, and perhaps it's kind of interesting, is it's a bit more of an anecdotal thing, but I had one developer, we had a bit of a discussion around biofouling, and when they scraped their device, took it out of the water, scraped all the biofouling off and put it back in the water, it actually got fouled more quickly. And so I guess potentially that's because there were either chemical or physical remnants of the biofouling. So they didn't scrape it all off potentially, there was something left there. And that served as a cue for biofouling animals that, hey, this is a great place to live, it's great habitat, to come and settle there more rapidly. So again, that's anecdotal, but I guess that's maybe something to think about in terms of cleaning procedures. And these are sorts of things that we really need to delve into the literature a bit more to think about, and potentially do some more experiments in the way that Jen's doing a lot of her work.
Great. We also have a question for Jen that notes that you explained the difference of fouling on the sides and underneath the Pelamis device, thinking in terms of light and depth only. And the participant wondered whether exposure and energetics could also define the differences you see in biofouling in different parts of a device such as that.
Yeah, thanks very much for that. It's an excellent question. That's actually my fault there for saying it was light and depth only. Sorry, I meant that those are two examples of the differences which we believe are helping to explain the differences in the biofouling. We're not sure exactly what it is that's causing the differences, and I completely agree that exposure and energetics is also something that could well be the variation between biofouling on the sides and underneath the Pelamis device. Unfortunately, we're never going to be able to say probably for certain exactly why it is that those two communities are different, because we haven't got the information measuring the exact, light through the year and the energetics and the exposure of those two different comparable areas on the device. But yeah, I think it's going to be a combination of a number of different factors that causes those communities to be different. And I just was highlighting their light and depth as a couple of examples. But thanks ever so much for that question. That's great.
Thank you. So following up the question that Rayanne answered about, and she noted that it was sometimes found that scraping the biofouling communities off a device or other item put in the water can cause sort of a renewed colonization very quickly. Can you talk a little bit about how far down would you have to go to scraping a device, getting it clean enough that you wouldn't get biofouling at an increased rate, in terms of what kind of slicks of bacteria and whatnot grow on these devices?
Thanks, Andrea. To say first off that this isn't necessarily my area of expertise, but in flippantly almost, I would say it's virtually impossible. But for example, a lot of work around current anti-fouling paints really focuses on some of the bacteria and the microfilms that you get, which are the very first things to establish on a structure literally as soon as it goes into the sea. Because that bacteria and those microfilms are really what kind of sends out a cue for all of these other plants and animals that this is a habitable environment. But with that said, looking on a slightly larger scale, things on the micro scale are a lot harder to fathom and practically maybe a lot more difficult to remove. But barnacles, for example, are gregarious, and what that means is that they're attracted to their own kind. So leaving behind barnacle shells, for example, any roughness, if you can clear all of that off and get back to a smooth surface, to me that would even be much, much better than leaving any kind of roughness. The other reason for that is that a lot of these creatures like to settle and land and live in areas that are rough. So for them, it's a bit of shelter. They might be, in the case of a tile device, for example, they might be protected from the extreme flows of the environment. So kind of being able to keep the surface as slick as possible might be, the best thing to do. But whether or not that's going to be practical, on the other hand, is a completely different issue, recognizing that, often these environments are quite difficult to access for people maintaining devices.
Wonderful. Those are all the written questions we have at the moment. If you'd like to just go to my last slide, please. I would like to very much thank our two speakers. I think this has been a really wonderful introduction to the concept of biofouling and some really interesting suggestions of where research and industry might go with the idea of helping prevent this from becoming a real problem to the wave and tidal industry. I wanted to note that recordings of this presentation will be posted on TETHIS in the next few days. I think that slide with the URL should be coming up. Not sure why it's not. We don't have another Annex 4 webinar scheduled at this point, but please watch for announcements on TETHIS and for your email. There will be another one in the fall. And if you're not yet on the webinar list, please send a blank email to join TETHIS webinars at lyris.pnnl.gov. We don't have that slide up for some reason. Anyway, go to TETHIS and you'll be able to see how to sign up. Meanwhile, I'd just like to thank our speakers again. This has been a great presentation, and we're looking forward to many more. Thank you.